Good day, grade 12s of South Africa. Today we are going to be working through genetics. Now, what is genetics? Well, first of all, it's a science. And secondly, it is the way we determine who we are, what we are, and how we are. I have a crooked pinky. I don't know if you can see it. This crooked pinky was handed down through lots and lots of generations to me. And if you have dimples, the shape of your nose, the shape of your eyes, the shape of your untweezed eyebrows, every single little thing about you has been handed down from generation to generation, and you are a combination of all your ancestors put together. Now, isn't that wonderful? You are unique. You are so special. And you've got a little piece of everyone that's gone before you sits there with you. You are a brand new perfection in totality. That's what you are. With genetics, though, please remember, Matrix, that we are only doing the tip of the iceberg. You are by no means a genetic specialist once you have completed genetics in grade 12. All right, so there are a lot of different variations and variables when looking at a person's genetics. So if you are slightly different to your brothers and sisters or your parents, it does not mean that you were adopted or you were found somewhere. It just means that there are lots and lots of different combinations, and we really do it so simplistically. It would be like doing grade 12 maths and only doing 1 + 1 is 2, or doing grade 1 arithmetic. You do a lot more than that, and that's what our genetics is all about. So let's have a look. Genetics, first of all, people, is a science. So it is a science without a doubt. Number 2, it is the study of the principles of heredity. In other words, what do you inherit as an organism, and also variation. Now, going back to when we did DNA and we did the nucleus and the way the nucleus works, we spoke about variations. We have genetic variations. Generally, that's when there is a slight mutation or a variation within your DNA replication. And when that takes place, if only one little gene is changed or altered, then the organism's reproductive process would also be altered, and the offspring that results will be different. And that links up when we do at the end our last module, where we do variation and we do diversity and survival of the fittest. It's because of variation that organisms change over a period of time. And that change over a period of time is called evolution. So please remember, you need to link your genetics and your DNA sections, the nucleus and DNA. Genetics gets linked to your section on diversity or your module on diversity, which you do the last quarter. All right, so what is genetics? It's the study of heredity and variation. And this you must know for the exams, because they will give this to you in a biological term. Now, DNA molecules produce specific proteins. All right, so we have specific proteins that are produced, and also it holds the genetic code. This is incredibly important. That genetic code is what you are. It is everything that you are. Every gene in your chromosomes or on your chromosomes, in your nucleus of every cell in your body. Now, how is that? There is a recipe, and that recipe is your specific genetic code. That's why you are special. So we've got our genetic code is unique to all living organisms. All right, any offspring will receive hereditary characteristics when the parents reproduce. Now, clearly you have parents. You have to have a male and you have to have a female to join during fertilization, the gametes to join, and then the offspring is produced. So haploid and haploid joins to form a diploid organism or a diploid zygote. And that's where you started out. Now, reproduce, and the offspring will resemble the parents, but also the grandparents and the great-grandparents. Why? Because your parents are a combination of all the others that went before. Genetic variation takes place because we have a crossing over during meiosis in prophase. Now, people, when we did meiosis previously, we went through that whole process of the crossing over in prophase 1. And that is where your genetic information is crossed over, so that your genetics are you, and you are a combination of your mother and your father, or the male parent and the female parent's genetic characteristics. If you were a plant, you wouldn't have a mother and a father. You'd have the male and female plants. Okay? And it ensures, this is incredibly important, the survival of the fittest. And this results in diversity. Okay? Without this, we do not have diversity. Alrighty, now, the role of gametes in inheritance, this is really just revision. It's revision of what we have done in grade 10, 11, and what we've done previously in grade 12. Sexual reproduction needs 2 haploid gametes, all right, to fuse. The haploid gametes are produced during meiosis. You know all of this, people. Gametes fuse during fertilization to form what? A diploid zygote. Remember, haploid was N plus N. When fertilization occurs, diploid, you end up with 2N. All right, now, DNA molecules on the chromosomes have the genes that contain what? It is the hereditary characteristics. Those sit on your genes, sit on your nitrogenous bases. Now, do you remember when we did the nucleus? We had a phosphate, a pentose sugar, and a nitrogenous base. And our nitrogenous bases were in complementary pairs. We had the purines and the pyrimidines. The purines, you had adenine and guanine, and the pyrimidines were cytosine and thymine, except in RNA, where you wouldn't have thymine, you would have uracil instead. All right, just to remind you so that we can carry on and you understand where everything fits together. Traits like hair color and your height and your body structure and your blood groups are all represented by genes on those 2 homologous chromosomes. Remember, homologous chromosomes, homo means the same. So they were the chromosomes for the same characteristics. One gene will come from the mother and one gene comes from the father, because you've got one chromosome will come from the father and the other from the mother. The genes the offsprings inherit will therefore be different from that of each of the parents. All right, that's exceptionally important. So you are going to inherit a combination from the male parent and from the female parent. You are then a combination of the 2. So if, for example, you have a brother or a sister who requires a kidney, who in the family would be the perfect donor for that kidney? It would not be the mother or the father, because the best they can be is 50% the same as the child. But they would look to the siblings, in other words, the brothers and sisters, because the chances of the combinations being similar in the other siblings are very close. It's very high, much higher than the 50% that you have between the mother and the father. All right, now, just to again refresh your memories, I'm going to use a highlighter here. We have the male parent would be diploid. In other words, they have a double set or a pair of chromosomes. The female parent is also diploid. Now, how do we go from diploid to haploid? Clearly, meiosis must take place. So meiosis will occur, and what do we end up with? We end up with a sperm cell, which is haploid, and an egg cell, which is haploid. And you must know this for when we do reproduction. All right, now, another process that occurs is going to be fertilization. In other words, fertilization when we have this sperm cell is going to join with that little egg cell. During fertilization, we then have our new little baby, our offspring. And our offspring is going to be called a zygote, and that zygote is diploid. It is 2n. So I have n plus n makes it 2n. All right, now 2 sets of chromosomes. I've got a double set of chromosomes again. Please, people, this is very important. You would have had, let's do it like this. You have one from the male parent and one from the female parent, okay, to make those sets of chromosomes. So we as humans have 46 chromosomes, or 23 pairs. 23 chromosomes for the same characteristics are going to come from the female parent and 23 from the male parent. And when they join together, haploid plus haploid in the gametes, fertilization occurs, and we then have our diploid zygote, which is you, it was me, etc. We are here, aren't we? So we were diploid zygotes that developed into an embryo. The embryo grew a little bit and became a fetus, and then the fetus, when it was big enough, birth was given to that fetus. All right, now we have mitosis, and mitosis is for growth and development of this diploid zygote, and we have our new offspring. And that new offspring then eventually matures and it develops into an adult that is diploid. And then, people, this whole process starts again. If this new offspring is a male, that side of it will happen. If the new offspring is a female, then clearly that side will occur. And then the whole process occurs again. And that is your cycle of life. Now, your important terminologies, please. And let's use our little highlighter pen. First, we have a locus. Now, locus is not short for locust. Locus is the location. So when they talk about a locus, it is the location of the gene on the chromosome. All right, it's a specific place on that chromosome will be where that specific gene sits. It's called the locus. So, and you have to know these terms, hey? All right, this is the exact position or location of a gene on a chromosome. All right, it's that gene, okay? What is an allele? Now, alleles, now look at the word, it's an A-double-L-E-L-E-S. It's a very easy word to learn to spell. Just don't get confused between all the L's and the E's, because it's A-L-L-E-L-E, an allele. Now, an allele is a pair of genes for the same characteristic on the 2 chromosomes. And if we look at the wording here, it tells you a pair of genes that are located on the same point. It's the same point on each of the 2 homologous chromosomes and represents a specific trait. In other words, and I'm going to do a little diagram here, a very simple one. There is one chromosome. This is the chromosome from the father, so it's your paternal chromosome. Okay, let's use the purple for the mommy one. So we have the mother's chromosome, there's the centromere, and this is the maternal chromosome. Now, sitting, I'm going to use bright green. Sitting at this point on the chromosome is the gene for hair color. And sitting on that exact point on the same chromosome, homologous chromosome pair, will be the gene for hair color or eye color. So, if we say that that gene is for hair color, and these 2 are our homologous chromosomes. So this here would be the allele, and that is the allele, so this makes up the allele pair. For what? For one specific characteristic or trait. Now you must think. You just use your head. You have millions of genes. Every single wrinkle, every dimple, every funny shaped bone, every bone, the front, the middle, the end of the bone, the shape of your nails, the shape of your cuticle, the shape of your nose. Now, I mean, if you look at your nose, you've got the bridge of the nose, you've got the side, you've got the top, you've got the nostrils. Every single little characteristic in your entire body has a gene that represents it. Isn't that amazing? You know, it's almost like you think infinitum, it carries on and on and on. That is how special you are, and every one of those genes is you and special to you. All right, so that's alleles. So we've got the locus and we've got the alleles. Now we look at homozygous. People, look at the word, and this is what we try to explain to you right in the beginning. Look at what a word is made of. Homo means the same. Zygous comes from zygote. Now you know, what is a zygote? It's when I have one gamete joins to the gamete from the one parent and the other gamete, and the two join doing a fertilization to form a diploid zygote. And that zygote will then develop into an embryo and a fetus and into you. So a zygote is the result. So homo, the same. So we have homozygous. It's what the offspring will look like, what the zygote will look like. Homo, the same, zygous. Homo means the same, zygous is the zygote. So when we have two alleles of a pair of genes are the same for one trait, then both alleles are for red flowers. The cross will result in a pure breed for red flowers. So I have one plant that has red flowers, and this plant is crossed with a plant that has red flowers. Guess what? What are the babies going to look like? What are the new plants going to look like? They are all going to have, and I can't use red because red's too bold here, but that would be red, and that crosses with red flowers. My result is going to be red flowers. Homozygous always means pure. It's a pure trait. In other words, homozygous for blonde hair, homozygous for black hair, homozygous for straight hair, homozygous for curly hair. Homozygous means pure. All right. Then heterozygous. If we have heterozygous, hetero means different. So hetero is different, zygous different. In other words, there are different characteristics that represent that specific trait. So here we're going to have different, and zygous means zygote. So when two alleles, remember the two alleles, the two characteristics of a pair of genes are different for a trait. In other words, one allele would be for red flowers, and the other allele is for white flowers. The cross will result in a mixture of the two genes. Okay, so homozygous means pure, heterozygous means a mixture. And I need for you to know these terms. So before we move on, a locus is the location. It's the specific point that a gene is found. Okay, alleles, each chromosome on the homologous pair of chromosomes in your body, you are going to have, let's say, eye color, and in exactly the same position on the other chromosome will be the gene for eye color. And because they are both genes for eye colors, we say that, at least eye color, we say they are alleles. Now, those alleles can be pure or they can be mixed. If they are pure, we say they are homozygous. If they are mixed, they are heterozygous, because hetero means different. Now, with mixed and pure and all the rest of it, we have what's called dominant traits. Now, if something is dominant, it dominates over something, and the opposite to dominant would be recessive. So dominant would dominate over something that is recessive, and it would stand back and almost cower in the corner. Dominant is always stronger, And that gene, that trait will always come through. Recessive will only come through if both genes are recessive. Otherwise, the dominating trait will dominate over the recessive trait. So let's have a look. Dominant trait, and here we say, we have a heterozygous offspring, so that's a mixture of the two parents, will display the dominant trait because it will dominate over the other recessive gene in the allele pair. Let's look at the example. If we have red color, red will dominate over white. So all the babies, unless they are pure, unless they are homozygous, they will be red. Even though they've got the white gene, they will still end up looking red. Why? Because the red dominates over the white. The same as black hair color dominates over white hair color, or brown eyes will dominate over blue eyes. There's certain characteristics that dominate over other characteristics. And this whole process here, this heterozygous cross, and that your dominant or your dominant gene will always dominate over your recessive gene, is actually Mendel's first law. It's called the law of dominance and segregation, and we're going to do that in our next session. So beware that you understand this, otherwise your next session's not going to make sense, and you won't be able to make sense of Mendel's laws. So we say, if we have the alleles always written with a capital letter if it's dominant, and in a small letter if it's recessive, when they give you questions in an exam, or your teacher gives you questions in class to do, if the letter representing that characteristic is in a capital letter, then you know it is the dominant characteristic. The recessive characteristic is always written with a small letter, and that is how we can distinguish the two. So if we said red flowers, red would be capital R, if it was homozygous red, in other words, pure red, and little r, if it was homozygous recessive. So this would be homozygous dominant, and this would be homozygous recessive. Okay? Now, if we have a mixture, it's going to be a mixture of the two, then it is heterozygous. It's a mixture of the two traits we are looking at. Okay, so to recap, and I need for you to understand this, that's why I'm laboring the point, is you have homozygous genes, which will be pure, and that pure trait can either be dominant, like black hair, or recessive, white hair. And the dominant, homozygous dominant would be pure dominant, homozygous recessive, pure recessive, and if I mix the two, I end up with heterozygous. Okay, so we have homozygous dominant alleles and heterozygous alleles. So let's look, you have to know this. Homozygous dominant means that both the genes are the same for the same dominant trait. Okay, so if RR represents red-colored flowers, then it means that both the genes are dominant. So we end up with two dominant genes present. Homozygous, the same, dominant, so it's right there, the same, and they are both dominant. So homozygous dominant, both are dominant. Okay? Heterozygous alleles means one gene is going to be dominant and the other is going to be recessive. So let's say we look at a flower and it looks red. Okay? It will be represented by capital R, small r, because we know it is not pure. This is pure. Here we know it is not pure. It is a mixture of the two. So heterozygous will always be a mixture of these two genes, the one that's dominant and the one that's recessive. And we then end up with, instead of here, capital R, or we could have had Yeah, okay, capital R, capital R. Here we have capital R, small r. It is a mixture of the two, which represents one gene for red and one gene for white. But when we look at that offspring, if it is dominant, it means that the flowers will all look red. But what's going to be inside their genes? It's going to be one gene for a dominant and one for recessive. So one for red flowers and one for white flowers. Okay, now this is just to show you what we could end up with. We have a homozygous dominant, so it's written with capital R, capital R. It's pure red. And here we have a homozygous recessive. It's for white flowers. It's written with two little r's. Okay, and we mix the two, and what do we get? We get a heterozygous dominant, so we have a capital R, small r, but these flowers will still look red. Why? Because red is dominant. Okay? So because red is the dominant gene, it will dominate over the white and almost cover the white up. Now we look at recessive traits. So if we look at recessive, dominant will dominate. Recessive is going to then be the weaker of the two. So we say the recessive allele is the trait that is dominated over. So it's the one that's almost suppressed. Therefore, suppressed by the dominant gene. It is always written with a small letter. Okay, now we get two types of recessive or recessive genes. When we talk about them, they are going to be, if we've got two recessive genes together, we say it is homozygous recessive, which means it is pure. Remember, anything with homozygous means it is pure. So homozygous recessive alleles means that both recessive genes are the same. In other words, our little gene for white flowers, both are going to be present, and only then will we see the white color. So in other words, people, if we have a dominance and recessive, all right, we look at homozygous dominant, we'll have both letters will be written with a capital letter. Let's say red color. If we have heterozygous, it'll be one capital and one small. In other words, it's a mixture of the two homozygous dominant and homozygous recessive. We put the two together and we end up with heterozygous. So here we have homozygous recessive. The only time we will see this trait for white flowers is if there are two recessive genes present. Okay, so the recessive trait, and this is incredibly important, the recessive trait will only ever be seen when it is in the homozygous state. So all the learners out there with blue eyes, with very, very light skins, with blonde hair, who are short, okay, those are all recessive traits. Okay, so you would, in order to see those recessive traits on you, we would have to know that we have two recessive genes. So on the specific locus on the chromosomes, homologous pair of chromosomes, the alleles are going to both represent the recessive trait. There are no dominant traits. Then it is homozygous recessive. Homozygous dominant, both the genes in the allele pair in the specific locus are going to be dominant for that characteristic. And if it is heterozygous, well then it's a mixture of the two, one dominant and one recessive. Okay, have a look here. Here we say homozygous dominant is our capital R, capital R. It's a pure red. And homozygous recessive, little r, and it's pure white, because homozygous is always pure. Heterozygous, if we have incomplete dominance, in other words, it is not complete, then we are going to end up with your capital R, small r, and we get pink. So, people, when we have dominance, we're going to get two types of dominance. One is going to be complete. So when it is complete, if I take capital R, so pure red, and I cross that pure red with pure white, I'm going to end up with capital R, small r, which is going to be red when I look at the organism. Why? Because I have complete dominance. But sometimes I have something called incomplete dominance. And when that takes place, and this is nice and easy for when you're doing any kind of genetic processes or calculations, because if there is incomplete dominance, then without a doubt, if I have the dominant times the recessive, I'm going to end up with heterozygous, but this heterozygous is going to be pink. So it would be the same as another example would be if I have black plus white and I end up with gray. Then I have incomplete dominance. If I take straight and curly and I end up with kinky or kinked hair. All right, so your incomplete dominance means that that dominant gene doesn't dominate completely over the recessive gene. In humans, an example would be if the one parent has brown eyes and the other parent has blue eyes, and a baby is produced and that baby has green eyes or hazel eyes. That is a mixture of the black, or at least of the brown and the blue. So when incomplete dominance occurs, then you're going to get a mixture of the two. They've got cattle that are red fur color and white fur color. And when they mix the two, you get cattle that have red and white patches all over. Your Friesland cows, if you know what your Friesland cows, they're good old dairy cows, the ones that give us all the milk, they're the best milk producers. Your Friesland cows are a combination of black cows and white cows. So you get cows with big black blotches and white blotches on their coats. They have incomplete dominance for fur color. Okay, people, now we've done incomplete dominance. We now go to co-dominance. And what is the difference? Remember, with incomplete dominance, you had red. I'm going to do it over here. We had red and plus white, and we end up with pink. Why? Because they don't completely dominate each other. One is it's incomplete. It doesn't actually happen. Whereas here with co-dominance, both gene alleles are equally dominant. So it's not incomplete, they both dominate. So the heterozygous individual will express both the traits. So if I have black plus white, in this case, just to use the same color, here we had red and white will give you pink. Or let's go red and white. So we do red and white. Red and white here will give you red with white markings, okay, that you sometimes see on roses. That would be co-dominance, whereas incomplete would give you pink. Now, examples, your most important, oh my heavens, please just, can we start again? Yeah, we'll have to. No, are we starting the whole thing, or do I start there? Is it going to be easier to pick up, or is it easier to start again? Easier to start again. Okay, sorry. My throat was going funny, and I was trying to avoid it, and I had to. All right, let's go again. Sorry, apologies. Okay, people, we've done incomplete dominance. We now go to co-dominance. Now, remember, with incomplete dominance, if I had red plus white, I ended up with pink. Okay? With co-dominance, if I have red plus white, I'm going to end up with red and white markings. Okay, like you would have on some roses. And that's the difference. With incomplete, it wasn't completely dominant. So your dominant gene was not completely dominant, and you ended up with a mixture of the two. With codominance, they will both dominate. So both traits will be expressed in the individual. The most important example you need to look at is your blood types. Okay, now remember, blood types are based on the proteins that may or may not be present. So if we have a protein A and a protein B, they'd be coded for alleles A and B. All right? Now, if no protein A or B is available or present, then it's coded for allele O. So A and B equally dominate, and O is going to be recessive. And these two are both dominant. And it's as easy as that. This means that every single time reproduction takes place, there are going to be 3 possible alleles. We're going to have A, we have B, and we have O. All right, so there are always more than 2 possible alleles, and because of that, we say it is multiple alleles. So whenever it is 2 or more, more than 2, we end up with multiple alleles. Now, any 2 of these alleles will occur in combination in an individual. So, alleles A and B, they are co-dominant. They equally dominate. And A and B completely dominate over poor old O, which, as I said to you, was recessive. Okay, now, when 2 individuals, for example, with blood type AB, when they have children, the children can either be A, or they can B, or they can be AB. Now have a look here. If I do the Punnett square, okay, and I say, right, here this parent is AB and this parent is AB, look at this. We end up with AA, we end up with AB, another AB, or BB. And that's going to determine what the baby's blood type is. Here, there would definitely be no O blood type. If there was, then we know both parents weren't AB. And that's what they use in hospitals to determine, very often they do blood testing to determine who the parent is not who the parent is. So, phenotype, if the phenotype, this is what is physically present in the blood cell, is A, it can either be AA or it can be AO, because remember, A dominates over poor little O. So that would be homozygous dominant, and here A would dominate over O. If we look at B, the same would apply here. We end up with B. If it's homozygous, we have 2 Bs. If it is heterozygous, B will dominate over O, and you will still see the B. If A and B are both present, then they are co-dominant, and we will see them as AB. And then shame, poor little O. O, as with all recessive genes, there has to be 2 O alleles for that characteristic to come through, because remember, it is homozygous recessive for the trait to be shown. Now we go genotype. The genotype, look at it. Look at the word. Geno comes from the word gene. So this is the genetic composition of an organism, and it has the information that sits in the genes. So for example, it will be the 2 genes on the homologous chromosomes for the homozygous dominant pure. They are heterozygous, or they are homozygous recessive pure. You cannot see this because it is in where? People, it is in the genes. So the genotype of an organism is what is written. It's the code. It's the recipe that sits inside the genes, on the chromosomes, in the nucleus of every living cell, except your red blood cells because they don't have a nucleus. But everywhere else, where you have a nucleus, this code, your genotype sits there. Okay? Can't be seen with the eye. Your phenotype here, pheno means physical. So the physical appearance of the individual organism. So what you will see when you look at the offspring. The phenotype is determined by the genotype, clearly, because the genotype is the recipe. And that recipe sits inside. It's like when you cook. Let's say you and I were going to cook dinner. I invite you to dinner. You're not going to see my recipe book, but you're going to see the dinner I've prepared for you. So my genotype in that case for that meal will be sitting in my recipe book, and you are going to see the proof of the pudding, which is what you are going to see just before you eat it. Sounds wonderful. I'm a bit hungry and peckish. I'm sure you are, because I know matric learners are always hungry. So here we go. Physical is your phenotype, this type of physical appearance, and it is determined, people, by what is written in the code, what is coded in the genes. You'll see white flowers for RR, but you will see red flowers if the genotype is capital R, or capital R, small r. In other words, homozygous dominant or heterozygous. Another example. And this is just so that you get the grasp of these terms, because you have to use these terms throughout in genetics. Okay, you must know your terms. If I don't like my nose, and I actually don't, but anyway, I've had to live with it for so long, I suppose I've got used to it. But this nose, if I go to a plastic surgeon and I have rhinoplasty, which means he gives me a nose job, all right? And he gives me this perfect little tiny ski ramp like Michael Jackson had, that beautiful little tiny nose. In my genes, the recipe for my big nose is still sitting there. But when people see me, they're going to see something different. Why? Because I've had a doctor change it. When I have children, my children are going to genetically end up with this nose. They're not going to have the wonderful little nose that the plastic surgeon gave me. Another example, let's say you have a very pale skin. Genetically, you have a pale skin. You have less melanin, because that's what makes our skins darker or lighter, is the presence of melanin. And by the way, the more melanin you have in your skin, the less chances, I mean naturally without tanning, the less chance you have of ever getting skin cancer. So the darker your skin, the safer you are, and the more protected you are from the sun. But anyway, we deter. Let's go back. You have a very fair skin, and you end up going to live in the Kalahari, or in the Southwest Africa somewhere, okay, and you're exposed quite a lot to the sunlight, or you decide to become a fisherman, and you're out on your boat in the sea fishing for hours and hours every day. You are going to get a wonderfully dark tan. Now, that tan is what people are going to see, but it's not what is in the genes. Okay, so please, people, be very wary. Genotype, what is written inside those genes. Phenotype is what you will physically see. If a flower or a plant produces red roses, it doesn't mean they are homozygous red. They could be heterozygous red, because the red dominates over the white. Okay, let's move on. We have our mono, means one, hybrid means mix. So we're now going to make our first offspring. Your monohybrid cross, mono means one, so when we have a pair of contrasting characteristics, it's crossed to determine the possible inheritance of the offspring. So there are always 4 possibilities. Why? Because you're going to get 2 possibilities from the male and 2 possibilities from the female. So how many chromosome pairs do we have? We have 2 pairs of chromosomes, people, 2 pairs. So 2 pairs of chromosomes from the mom, 2 pairs of chromosomes from the dad, and we put them together. So this would be our combination A, our combination B, our combination C, and our combination D. And this is true for every single time reproduction takes place. Whenever reproduction takes place, we are going to have 4 possible combinations. In other words, if the chances are let's say this is X and that is, or this is X and that is X, and this is X and this is Y. We're calculating the number, what the chances are to have a boy. All right? So we go here and we say, right, A would be X, X. B would be X, X. C would be X, Y. And D would be X, Y. So there would be a 50% chance of producing boys and a 50% chance of producing girls. But for every single time reproduction takes place. So every time that happens, for every characteristic, you're going to have 4 possible combinations. It results in 4 combinations every time. Now, if we look at a dihybrid cross, di, as you know, means 2. So when we have 2 pairs of contrasting characteristics, it says that there are 16 possibilities. Those 16 is your 4 times 4. So you're going to have 4 from the male and 4 from the female possibilities or combinations which result in the new offspring. And that applies to Mendel's second law, okay, which is the law of independent assortment. And what we say there is, if the parents have hair color black and white, and eye color is brown and red, because you're working with guinea pigs, for example, you've got any of those combinations that will occur. So black with red eye color, black with brown eye color, white with red eye color, white with brown eye color. It gives you 16 different combinations. Okay, now we carry on with our definitions. Filial generation. Filial means the first group of children. It's your first family or family. So filial, think of the word, it's not the direct translation, but think of the word as the family. So P will be the parents, okay? And the parents are now going to have the F1 will be the first family. Then these little babies become the next family group, and then they will produce their babies, which will be F2. And that's what all of this says. The parents are represented by P1. The parents reproduce to produce offspring that result from a cross. So the parents cross. The offspring of the first filial generation, which is F1, is represented like that. When those offspring now mature, they reproduce, they become the second lot of parents, and they will then be the P2 generation. Their offspring will then become the second filial generation. Now, how many of you can actually, this is quite a nice exercise to do when you have time during the holidays, instead of lying in front of the TV watching soap operas. Talk to your parents, talk to your grandparents, talk to your aunts and uncles, and see how far back you can trace your family tree. It's actually a science, and it's fun, and it's wonderful as a hobby. And you can go back and you can see your great aunts and uncles and your great-great-great-grandparents, and they would then, as far back as you can go, they would be P1. And their first children would be F1. And then those children become parents, so they would be P2. And their children F2. And so you can go through generations until you end up with P55 and F55. All right, so it's quite fun. And I'm sure you're going to say it's raw, sounds like great fun. It is, I promise. It's also very interesting to see where you come from and what your background is. So, Punnett square. What is a Punnett square? It is a way to represent a cross. So we use this to show a genetic cross. And it's a nicer way of doing it. Another version is to write the genes up like this, and then this one combines with that one, and this one combines with that one, and this one combines with this one, and it ends up being a lot of little lines, and you end up very, very confused. I don't like using this. I find that learners get less confused when they use a Punnett square. Now, in an exam, they will specify use a Punnett square, or they will say do a genetic cross. They will never ever tell you to do the line version. So they will either specify to use a Punnett square, or you can use any process you like. However, if they do specify Punnett square, if you use anything else, you get naught. So just do it the Punnett square way and it becomes far easier to understand. So we now go to our Punnett square. And this is what we have. Here is an example where the one parent is capital B, and the other parent is little b, little b. In other words, this one is going to be homozygous dominant, okay? And this one is going to be homozygous recessive. All right, you have to write this out. I'm not writing it out. I haven't got time. Otherwise, I'm going to waste time when talking to you. Okay, so what we're saying here is the capital B is for brown. In other words, it is dominant. And blonde is recessive. And if both those parents are capital B, and small b, they are homozygous dominant. All right, here we go. And this is how we would do our square. And what I've done here is I've shown you where your mark allocations are. So this is very, very important. Okay. First, you are going to write P1. And your parents are going to be here. That's what your parents are. Okay. So we say there they are. Now we have a tiny little problem because this here I saw now should actually go here. This is where our first offspring is. So the P1s are going to be here. So with P1, we have our gametes. The gametes are going to undergo meiosis. And in this whole process of reproduction, this is what we're going to sit with. Capital B, small b, capital B, small b, capital B, small b, and capital B, small b. So you're going to get a mark for P1. You get a mark for F1. You get a mark for putting this down. You get a mark for these gametes and a mark for those gametes. And then you get 2 marks for representing these 4 correctly. So they literally count half each. Now, your genotype, that is what you are going to see in the genes. This is the recipe written in the genes. There are going to be 4 out of 4. You see there are 4 blocks? So 4 out of 4 are going to be capital B, small b, and they are heterozygous. They are a mixture. And for this, 2 marks because you get 1 for the ratio and 1 for what it is. Always include the ratios. Then phenotype. This is what we are going to see physically. Now remember, if we have complete dominance, you are always going to see the dominant trait. So this would be 100%. 4 out of 4 is 100% brown. 2 marks. And always include the percentage when you do the phenotype. So for the genotype, you have ratios. In the phenotype, you are going to have percentage. Now, let's see how you have absorbed everything I've taught you so far. And we're going to do question 1. Now, this question 1 was taken from the National Senior Certificate Examination in November 2008. And I've given you the question number here. It says here, people with albinism. Now, this is how you're going to answer a question in an exam. You may think this is the long way around. It isn't, because what it does is it says to your brain, okay, pause. Let's check this out. Pull out the files that are relevant, and then we take those files, and then we can answer the questions. Okay, so what you're doing is, it's like if you were going to run a race. You're first going to warm up and prepare your body. And once you've warmed up and you're ready, and then you go and you do your race. You don't just dive into a 100-meter sprint from having just gotten up at home and you make your way to school and then all of a sudden you do a 100-meter race. You warm up first and you get psyched up and you get your head ready. That's what you're doing here in an exam. And it's not silly to do it. You need to warm up. Otherwise, you could never run a 100-meter race at full capacity and do your best. Why? You didn't warm up properly. You need to get the brain ready. Let the brain know this is what's coming. So, exam technique. First thing, you read the statement that they give you. So we say, people with albinism are unable to produce dark pigment. Okay, that's fine. So they're missing melanin in their skin. And a lack of melanin means that they can't tan properly. They can't produce the dark pigment. This condition is caused when an individual is, now please, homozygous recessive. In other words, we're going to have two little letters for this characteristic. Now, the family tree below shows the occurrence of albinism over 3 generations. So let's have a look at it. Now remember, albinism is the ability or the lack of the ability to produce a dark skin color. So what have we got? We got grandparents of the P1, and then there are the babies. Now remember, this F1 is going to become P2, and F2, if they went to the next generation, will become P3, and so we carry on. So filial generation is the offspring. Here we have a man without albinism. Here we have a woman with albinism. Here is a woman without albinism and a man without albinism. So you can't see it. So what they're showing you here is what is in the phenotype. And the child without albinism, but a child with albinism. So your first question is, indicate whether each of the individuals below could be homozygous dominant, homozygous recessive, or heterozygous. So what we're going to do first is, if in an exam you are given this. Now sometimes they put circles here and little squares, and they indicate it with circles and squares, and then they'll give you a key. That is called, at least you have your key here, and this whole process here tells you exactly who does what. So we are going to say for albinism, we're going to make it a capital A. So the A will represent the dominant characteristic. So this will be no albinism, because that's what dominates, because they tell you it's a recessive trait. And the little a will represent a person with albinism. So let's have a look. If this person is without albinism and this woman has albinism, she's going to be little a, little a. She's going to be homozygous recessive. All right? And here, the man without albinism, we don't know what he is at this point. But if he hasn't got albinism, he must have a capital A. So we know definitely he has a capital A. We don't know whether he's homozygous dominant or whether he's heterozygous yet. Now, we've got the woman without albinism. Again, we don't know what she is. But man without albinism. But now look at this. This woman and this man without albinism produces a child with albinism. And we know for that child to show albinism, it must be homozygous recessive. So this child here can either be capital A, or capital A, small a. In other words, homozygous dominant or heterozygous. Here, the woman without albinism and the man without albinism, the fact that they produce a child with albinism means they must both be heterozygous. And if this, these parents here, they don't have albinism, but we know the child they produce doesn't have albinism, but that child must have the gene because, look, the mother was an albino. So the father could be that, or the father could be heterozygous. Because as long as one of these A's goes here, let's do it like this. As long as one of these A's, whichever A it is, ends up there, because the little a is going to come from the mother. We know that already. All right. And that's their offspring. So the father could be homozygous or heterozygous. So what they wanted from us here is indicate whether the individuals below could be homozygous. I think A is 1, so we look at 1. Could either be homozygous dominant, in other words, AA, or heterozygous. And heterozygous means that they would be capital A, small a. Remember, the capital letter is always used to write the dominant trait, and the little small letter is used to write the recessive trait. And when it is heterozygous, it will be the capital letter and the small letter. All right, now for B or 2 would be this child here with albinism. It has to be homozygous recessive. All right, and what are we doing? Here we are looking at the genotype. Now, it says here, explain your answer to question 1.1a. In other words, why did we say that that father was either homozygous dominant or heterozygous? And we say this. We say that the normal is dominant, because normal is dominant. Normal would be not to be an albino. So normal is dominant, and the dominant condition or dominant trait or characteristic normal is dominant, and the dominant characteristic can show as either homozygous, clearly dominant, homozygous, or heterozygous. Okay, and that's all you have to say. So please remember that your dominant characteristic, if you can see it in the phenotype, it is either homozygous dominant or it can be heterozygous. And if they produce an offspring that is heterozygous, well, it means that somewhere they got the recessive gene from the mother. The father could have had a recessive gene, because in every cross there are the 4 chances, every single time. So if you have, let's say these people had 4 children, none of them may end up having albinism, but all 4 may also end up having albinism. Each time fertilization occurs, okay, every time fertilization occurs, those 4 chances come into play, every time. Okay, now, okay, people, let's look at question 2. Now, this was taken from the National Senior Certificate Examinations. We're looking at November 2009, and I've given you the question number as well. Okay, here we've got fur color in mice, and it's controlled by a gene with 2 alleles. So they give you this information, and they say, right, we're looking at fur color in mice, and it's controlled by a gene with 2 alleles. Most genes have 2 alleles except for blood groups. Your blood groups have 3 different alleles, so we call that polyploidy. There are 3 alleles that will determine blood group. But for just about all the other characteristics you will do at school level, you will only have 2. So, or multiple alleles. Here we only have 2. So with 2 alleles, okay, we have a homozygous mouse with black fur color was crossed with a homozygous mouse with brown fur color. Okay? All the offspring have black fur. Now, the fact that they've told you this, they've said to you all the offspring have black fur when you cross homozygous black with homozygous brown, immediately you know, before you even read on, you are going to know that black fur is going to be dominant and brown is going to be recessive. Immediately, okay? And any homozygous and homozygous crosses together will always give you heterozygous offspring. So, it says here, using the symbols capital B and small b to represent the 2 alleles for fur color, show the diagrammic representation, in others we're going to use a Punnett square, of a genetic cross between a mouse with heterozygous for fur color with a mouse with brown fur color. Show the possible genotypes and phenotypes of the offspring. Okay, so they've told you here what controls it. Okay, and if we have a homozygous black crosses with a homozygous brown, we are going to end up with all the fur in the all the mice in the heterozygous state. They are going to have black fur, even though they are heterozygous. So now you know black is dominant. Okay, so black represent the two alleles of fur color. This here is something that they're going to expect you to know to apply in your life science. Remember, it's not always regurgitating what you know and quite literally just spewing out the information. One of the major skills we are trying to teach our learners at school is to be able to apply the knowledge you've learned. It doesn't help if you know your 5 times table off by heart, and I'm using a very silly childlike explanation here, but it doesn't help if you know the 5 times table off by heart. If I give you 25 cents and I ask you to divide it between 5 kids or 5 children, are you able to do it? And that is what we're looking at here. You're going to have to know your content and then be able to apply that content in a context here. They're going to give you problems to solve, and you're going to have to know your work to know which principles you have to apply. Okay, so using the symbols capital B and small b, we've already established that. We're now going to show genetic crosses between a mouse that is heterozygous, so capital B, small b, for fur color with a mouse with brown fur. Now, if that mouse has brown fur, and we can see the brown fur, clearly we can see it, therefore, this little mouse is going to be small b, small b. It has to be homozygous recessive for us to see the trait. Okay, people, please remember that. And they've used black and brown hair color here. They could have used black and white. They could have used brown and white. Okay, but they've told you very clearly here, when you have homozygous and you cross them, in the heterozygous state, the fur is black. So there they told you indirectly, black is dominant. Okay, so homozygous, at least heterozygous for fur color with a mouse with brown fur. In other words, little b. So, and they want us to do the genotype and the phenotype. So now, here are where your marks are. P1, what have I got? This is my first parent set. We know the parents are capital B, small b, heterozygous. Little b, homozygous. Okay, but homozygous, recessive. Okay, we also know that B is for black, which is dominant. And we know that the little b is for brown, and this is going to be recessive. Okay, now we get clever. We say, right, let's do our cross. First, we've got our gametes. These are our gametes, and the gametes are going to undergo, this is after meiosis has taken place. We end up with one chromosome will have the B, and the other chromosome possibility would have the little b, and so we do it. Now, capital B times little b times little b, capital B and little b, and small b, small b. All right, that is our P1 generation. And our P1 generation, we had our capital B, small b, and our small b, small b. Heterozygous, homozygous. Now, we go to our F1, or first filial generation. And our first filial generation, the genotype. Okay, now with your genotype, we are going to look at our ratio. Remember, genotype always has a ratio. So we're going to have 2 in 4, because I've got 4 possibilities, 2 and 4 are going to be capital B, small b, which is heterozygous. Okay, in the phenotype, this is going to be what I physically can see. I'm going to have 50% of the offspring are going to be black, and 50% of the offspring are going to be brown. Okay, and there it is. It's as simple as that. Now that wasn't complicated, was it, people? It was actually very easy. Remember P1, F1, and your gametes, fertilization takes place, and those are your possibilities. Let's just quickly recap here where your marks are going to be, because that's after all what we are interested in here. So we're going to go P, our gametes and meiosis, and we have those two and those two, and you get 2 ticks here for your combinations. You get 1 for F1, 1 for writing genotype, phenotype, your ratios, your percentages. Those are the maximum number of marks. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. This could count 16 marks. They could also make it count 8 marks. So that they give you 2 marks per tick. Okay? Right. Now, I'm feeling confident that you know what's going on. So let's look at question 3. What is the genotype of a plant that results from a cross between a plant with red flowers and a plant with white flowers? Now we're looking only for the genotype. They're not asking for the phenotype, eh? Only the genotype. So I think let's use the blue because I think it stands up nicer. So we want to know a cross, so we're going to say red, and they've told you dominant because it's capital R. So red is capital R, and white is small r, and that already tells us it is dominant. Now, what do we have? Our gametes, we're going to have R times R. Those are our combinations. P1, I need to just make some space, P1, and in P1 we have our Punnett square. Here we have our gametes, and the gametes that are going to result after meiosis has occurred. Let's put a little arrow there. Okay, capital R, small r, and this was our rule. I told you, whenever you have a homozygous dominant crossing with a homozygous recessive, you are always going to end up with the offspring that are heterozygous. So we now go F1, and what are we going to have in our F1? Now all they wanted from us was the genotype, okay? That's all they asked in the question. The genotype is going to be 4 to 4, because we always do the ratio in the genotype. Capital R, small r, which means they are all heterozygous. Okay, and that's quite easy. But what I'm going to do here, just for practice, I'm going to write the phenotype. Now remember, phenotype is what we see physically. Okay, here, percentage, we're going to have 100% of the organisms are going to, what are they going to look like? Red was dominant. They are all going to be red. Okay, now, if we have incomplete dominance, that's always easy. If you get a question in the exam for with complete dominance, you say, thank you, because if it's complete dominance in its homozygous dominant state and heterozygous, you will see the dominant trait. But if it is incomplete dominance Well, red and white will give you pink. Black and white will give you gray, or black with gray with white spots on it. So it's always the in-between color. Okay? So here we go. We have 2 white heterozygous cats. So they are white heterozygous cats were crossed where white fur is dominant over black. Now people, if you recall earlier, I said to you, black normally dominates over white, and red will dominate over white, and black will dominate over, or brown eyes will dominate over blue eyes, black hair over blonde hair, unless they tell you otherwise in a question, and they've just done it here. They've said, hold on, we're going to tell you now, white fur dominates over black fur. So white is going to be dominant. So let's do it like this. We make capital WW, or W is going to be for white. Although I don't like using W's, but it's what we stuck with here because of the white fur. And we're going to do a little w is going to be black fur. And we say, write down the correct genotype and phenotype for this cross. So be sure, I've underlined, you can make little notes here for yourself. Do whatever you want to. Your exam paper is yours to write on. Okay? So we've got 2 heterozygous cats, which means they are a combination. So we're going to say we have the parents, so P1, and our parents are going to be capital W, small w times capital W, small w. All right? That's our cross. So our Punnett square, and we have gametes and meiosis. They undergo meiosis. And we're going to have W, little one, W, little one. So 2 capitals, a capital and a little, a capital and a little, and a little and a little. My F1 generation in the genotype, what ratios do I have here? I have 1 in, sorry. I'm trying to finish off here because we're soon done with our lesson. So here we would have 1 to 4. And 1 to 4 would be capital W, so therefore homozygous, dominant. Okay, and we would have 2 in 4 would be capital and a small, that's heterozygous. And we end up with 1 in 4, little w, and that would be homozygous recessive. Please, you need to write these out. I'm running out of time here. So people, that is pretty much in a nutshell our terminologies that you need for genetics. Make sure you go through it. Make sure you look at your X sheets because they're all in your X sheets. So check them, make sure you know them. You can get them as multi-choices. You can get it in the section on column A, column B's, or provide the correct biological term for, or within an actual question. But you must know those terminologies. Homozygous recessive, homozygous dominant. What is the phenotype? What is the genotype? You need to know that in order to do your questions and get the maximum marks for your questions. So there you go. We come to an end for this session. Our next session, we're going to look at blood groups, we're going to look at sex determination, and eventually we're going to do genetic diseases, and then we move on to reproduction. But you must know this so that you know what the babies are going to look like. So have fun. Until I see you again, have a good one. Bye-bye.